Thank you very much for coming to another seminar. Today we have Enrique Sánchez-Fabres, a very long surname. He's a master student in the Mares group in the fish ecology section, and he has been working. I think he has had one of those high intro scholarship that is funded by the María de Maestro.
Perdona, és que estem sentint en estèreo, se té algo que no has bloquejat.
Ah, vale, no, aquí a veure.
Perdó.
No, millor arreglar-lo ara, no? Pues aquí,
Sí,
a veure,
vas a
YouTube
arreglar-lo.
està...
T'estic sentint també de
Vale,
sortir.
aquí quita el sonido. O sea, ¿tienes el sonido quitado?
¿El micro? ¿Y el micro?
Ah, no, ese.
No, pero lo tenía en silencio.
Sí. A ver, ahora.
¿Ahora?
Hola.
Sí, sí, oyendo.
¿Tenéis algo conectado?
Hola. Sí, está aquí.
No, tienes el sonido quitado. Ah, no, tienes. Sí. Hola.
Yo creo que ya.
¿Qué? Ya no estoy en bucle.
Que te sentía, te oía doble.
Okay, so as I was saying, he has been working with one high intro scholarship that is funded by the María de Maestro Award, and also in his case by the Arenal Nautic Club. So that's his funding part of his work here. And he will explain his master project. So the floor is yours, speak.
Thank you. Hello. So, yes, I'm Enrique, and that's for names for those who don't know me. And I'm going to show you the work I've been doing since March with the high intro IQ that was sponsored by the Arenal Nautic Club. And well, the title I put here is not the same that I sent, sorry, but well, basically it's incorporating individual behavior on population dynamics of exploited fish through an individual-based model. So I will start giving you a bit of context of the study and what's the motivation behind. I will try to transmit the objectives, and then I will show you the methods, how the model works, and how we implement these behavioral traits in it, and then the results and discussion that we can obtain from it, and some conclusions and future steps as well. So the first thing that you need to know about the context of this study is that the fish have different behaviors within the individual level. So one fish behaves different from the other. This is something maybe not that surprising, but it's something that is been able to study relatively recently, thanks to methodologies like the acoustic telemetry. So this is being the case for the pearly razorfish or raor, a very important species here in Balearic Island. It's been studied a lot with this methodology, and here, for example, in the group of PEP, it's been possible to observe, describe different behavioral types at the individual level. And we will center specifically in two of them, the home range and the chronotype, that I will explain later what they are and how we implement it in our model. So the first thing I wanted to also describe is about the acoustic telemetry. For those who are not familiarized with it, it's a methodology to try to study the spatial ecology of the fish in this case. It consists in the use of receivers like this here. They can be in a mooring or in a buoy or wherever you want to put it, to try to record the presence of fish or other species, individuals that you previously have tagged with acoustic tags that can be external or internal. So the idea is that these tags emit a radio frequency that when it is a certain distance from the receiver knows and record the presence of the animal. Well, as part of my fellowship, I had the chance to work in this, in all that involves this kind of methodology. For example, doing concrete to make the moorings. I learned to do concrete. Someday maybe I can do my own barbecue, which is nice. So the results were very good for scientists as well. And these are the moorings. This is an example of the moorings to put the receptors, and you have here an example of the receivers. Sorry, the receivers that are used in acoustic telemetry and that we use to study the spatial ecology of marine fish and coastal fishes in Balearic Island, for example, which is from the Balearic tracking network. So I also had the chance to go to the sea and put this in the water, throwing it everywhere, or well, no, in specific places, especially design, following methodologies and all that stuff. Sometimes we also took them back, obtain and download the information, and then put them back in the sea. This is the same. So our study case, our subject species, as I mentioned, is the raor, pearly razorfish. Xyrichtys novacula. Well, this is a particular species and very interesting for studying in acoustic telemetry because it has some traits like they inhabitate in sandy bottoms. They are very territorial and they have a REM-like structure and social aspect. And they also burrow in the substrate. They live in sandy substrates and they burrow in the night to sleep or to avoid the predators. So as I mentioned, these traits make them an ideal candidates to study with acoustic telemetry. And I also had the chance to go to the sea and do some tagging campaigns and fish myself some raors and tag them, and it was very funny. So the studies that have been developed around this species and other in more general perspective of marine and fish have allowed to observe the existence of spatial and temporal behavioral phenotypes, that different individuals can present. So this can also be translated as that they have different personalities. So these different personalities will imply that they have a variation in the movement at each individual, and is what brings that there exist different behavioral types. For example, the home range and the chronotype that I mentioned before that are going to be the ones that we will focus on. And the idea is to incorporate these behaviors in our model at the individual level. So to explain what is each of these things, the home range is pretty straightforward. It's basically the area that the individual uses in their normal activity. So you can see here an example of a study done with pearly razorfish and acoustic telemetry. And in the y-axis, you have the area in square meters, the territory size, which is exactly the same that home range. And for example, centering only in the males, we have 2 seasons, and each point corresponds to one individual. And you can see here that different individuals have different home ranges. Some are bigger than others, and they are quite well. Some points you can see they have like mustache, which is like the variability of this home range. The general thing that you can observe here is that the home range is within the individual level is pretty constant. It's not very variable, and even between different seasons, like pre-reproduction or reproduction, it remains also pretty constant. So this brings the idea that different individuals present different home ranges, and it is a good trait to observe and to try to incorporate it to our model. The same with respect to the chronotype. The chronotype is the circadian rhythm of the individual. It's like the periods they have for activities and for resting, and at which point they pass from one activity to another. For example, the awakening time, so the time that they wake up, is a good metric to measure or to characterize at the individual level what is their chronotype. And what we can see here in this study from 2018, that some individuals wake up, this is the sunrise, so we start counting minutes once the sun is out. Some individuals wake up, well, with the sunrise, some a bit later, some a bit later. And we see some individuals that wake up to 2 or 3 hours later than others. So this is a big difference. It's like humans, some just like to wake up early and some later. And in this plot here, what we can see is that each of these violets is one individual, and their variance in the awakening time. And we can find some individuals that really remain in the same time of awakening time. So there will be a difference in this behavior at the individual level that will have also implications in for example, the vulnerability for the fishing pressure or other circumstances of the environment that I will explain a bit later, how we use these chronotype and home range traits to make some ecological assumptions with respect to our model and the fishing pressure. So for now, the objective is to investigate the implementation of these behavioral traits in a population dynamics model for the species, for the RAOR, and the way is developing an individual-based model that incorporates specifically the home range and chronotype. So now I'm going through the methods. I'm going to start with a very simple formula that will not scare anyone because it's just the model, our model, and generally speaking in fishery sciences, the biomass, the models that describe the stock of the population are based in biomass, so the weight of the overall population. So this formula describes what conforms the biomass of a population at a given time. So we start with a population in the time t. They have a certain biomass and they grow, so they gain biomass. So you add biomass to the population. Then they can reproduce, so they will be recruits that get into the population. So they will be, again, a gain of biomass. And then some individuals will die by natural causes, natural mortality, or by fishing mortality. And there will be then a loss of biomass. So this is the main model, okay? This is all you need to have clear to understand how the population dynamics works in our case and in what we are doing. But behind each of these parts, there are more parts, okay? There are a lot of formulas describing it, especially in the part of the mortality, because it's the model that we have put more effort in characterize the influence and the effect of the individual behavior in the different probabilities of dying by natural or by fishing mortality. So you don't need to go into it, just for you to know that behind that easy formula, there are more things. And if you want to go after in the questions, okay. But so I'm going to describe the model very in a illustrative way for you to understand how it works. And it's basically the same, following the same path that the formula that I showed you for the first one. We have an initial population of fish, flowers there, and they have a given biomass altogether. And each of these flowers have their own traits. They have an ID to identify them, age, length, weight, home range, chronotype, and growth rate. So specifically, the home range, the chronotype, and the growth rate, that it's K, is the factors that bring heterogeneity among individuals. No? It's what characterizes each of the individuals. The length and the weight are dependent of the age, also are dependent of K, of the growth rate. And we have also make the growth rate dependent of the home range. Now I'm going to explain a bit the ecological assumptions that we make to implement the effect of these traits in our model, specifically in the model of mortality by fishing or by natural mortality. So based on the works that I showed you before, and more other works that show that have shown that individuals with, for example, with a bigger home range will have a higher vulnerability to fishing, for example, because if you have an individual that has a bigger area than another, and you have a certain number of fishing boats, for example, fishing in that area or in a wider area, the individual with higher home range that will move in a more extensive area will have more probabilities to encounter one of those fishermen or one of those boats. So the idea is that a higher chronotype, a higher home range will imply a higher probability of being fished. The same with natural mortality, higher home range, higher probability of encounter a natural predator. So with the chronotype, it's a bit similar, the process. Those individuals that are awakening earlier, fishermen are known to be to wake up very early, to be there in the fishing ground with the sunrise and not losing any minute of light. So fishermen are going to be there. Imagine like this, okay? It's an example, and it is based on a study and the simulations that have been done for this species. And what they show is that if the individuals wake up early with the sunrise, they will have their activity will overlap with the activity of the fisherman more than if they wake up 2 hours later, then maybe some fishermen have left or they have already fished other fish. Whatever the way, the case is that a higher chronotype in terms of higher means that they wake up earlier will mean also higher probability of mortality. And then we also introduce the home range as dependent, the growth rate as dependent of the home range under the assumption that an individual that is going to move more and have a more extensive area will also get more food and will grow more. Okay, so these are the assumptions behind. And then some individuals live and some die by fishing or by natural causes. And the survivals, the next step in the model is to apply the growth model, and that depends on the growth rate and the age of the individuals. So they grow, they become bigger, as you can clearly see. And then the survivals update their traits, so they are 1 year older and they are bigger, heavier, and these remain the same. And these will be able, they have survived, so they can have recruits that are added to the population as well. And these new recruits, it's important that they will get their home range, chronotype, and growth rate traits from the pool of traits of the spawners. So this gives the model a factor of heritability that will be also interesting to observe the results later. And this is our population after 1 year, one time step. In the case of the model, it works in year. So after 1 year, we have this population that have gone up through all this stuff, and then we start again, and we could do projections in, for example, of 100 years. So as you see, this is the formula that I showed you at the beginning with the colors. Okay, so I hope it's more clear for now. Good. If you have a question now, it's a good moment, because then it's going to be hard to stop. Okay? Okay.
I wonder the
Chronotype?
the chronotype, how does it affect the natural mortality?
For that, the assumption could be similar. If they wake up, if they wake up later, they will have less period of activity overlapping with other predators. Because when they are sleeping, they are technically safe, unless one dolphin comes and. But yeah, that's the assumption.
Because the predators are also early people.
They are assuming that they also are active in the daylight. Or independently, if they are borrowed in the sun, it doesn't matter how is the predator doing because they are safe. Okay, so now that you know the model, we did different simulations in different scenarios. We wanted to compare a population with individual variability, where all these factors of home range and the chronotype and the growth rate as well are incorporated in the individuals. Okay? Comparing with a scenario where there was no variability in this trait, so all the individuals had the same scores for these traits. And we wanted also to compare different fishing scenarios to see how the population with variability or with no variability, they respond to different scenarios of fishing effort that we had from A1 to A6. The higher the number, the higher the effort. Okay. So the effort in the fishing, not going too much in the formulas, just is we could parameterize it with this factor here. So the idea was to compare different fishing efforts, how they could affect the populations, and comparing with the effects of the individual variability or not. So now we can go to the results. The first that I'm going to show you is the biomass and the yield. I'm going to explain now what is that, trends under the different effort scenarios. So what we have here is the biomass of the population. And remember the formula, it's all defined by the overall biomass. How it is changing along 100 years in these two scenarios. Now we have variability in blue. So there are different individuals with different characteristics, and with no variability in red. So what it's easy to observe here is, in general terms, that the population in the scenario where there was variability in the population, the general biomass, the trends of the biomass are higher than the trends in the case of the population without variability. Up to 50% more biomass, so the population was 50% bigger comparing to a case where there was no variability. And the yield is the amount of fish that is caught, so how much it is fished, no? In terms of kilogram, that's the yield. It is also possible to observe that in the scenarios with variability, there was more yield, so it was possible to fish more than in the cases of no variability. This was a bit more variable, but from 25 to 70% more biomass of fish that the fishermen could catch, no? Theoretically speaking. So you see this, okay? And of course, in different scenarios of different fishing efforts. So here is the lowest effort, and here the 6 is the highest effort. It's also possible to appreciate that the higher the effort, the overall population is smaller since you are fishing more. And the fishing, the yield also tends to be higher, but it's not linear correlation, because if you fish too much, if you're fishing a lot, you're doing a lot of effort, you are also decreasing the population. And if you have a small population, you will not have a lot to fish. So it's a process that is not linear, and it is explained by what it is called the maximum sustainable yield. The maximum sustainable yield is the maximum fish that you can get to guarantee that your population is going to remain stable. So it's not going to decrease, it's going to be stable. So you are going to be able to keep that exploitation rate indefinitely, theoretically speaking. So it is defined here, it's very easy. So the higher the effort, you are going to catch more until you get to a certain point, a threshold, from which your population is too exploited and they are not going to be able to reproduce at the same rate. So there's going to start to be a decrease of the population. So the maximum stable yield in fisheries science is like the concept that is used to try to manage the exploitation of the stocks and try to do it in the sustainable way, but also the more with the most outcome possible. And with this concept of with this concept of maximum sustainable yield, there are another related concepts as the biomass at the maximum sustainable yield. So this is the biomass that your population needs to have to give you the maximum sustainable yield. And there is also the mortality rate or mortality pressure at the maximum sustainable yield. That is the same as the maximum effort that you can do so your population stays stable and you get the maximum outcome in a sustainable way or in a permanent way. So I am explaining all of this because the next results lead to this background. And the Coway plot that I show here is a way to basically represent the status of a stock based on these concepts. The idea is to bring you the state of the given stock in terms of how is their biomass and how is the fishing effort they are suffering, and how is this affecting to the stock in the terms of if it is healthy and it is even growing, or it is being overfished and it is already overexploited, so it is under the biomass of the maximum sustainable yield. And there are like tangential cases in which your population can be over the maximum sustainable yield biomass, so you have more biomass than you need to get this yield, but you are fishing too much. So if you keep that pressure, eventually your population will start decrease and can go below the ideal amount of biomass that brings you the maximum sustainable yield that would be here in the intersection. And the other cases that you can have that I explained would be this scenario, and then the last scenario and the other scenario is when you have a very low or have a population with a biomass lower than the biomass you need to get the maximum sustainable yield, but you are not fishing in a high rate with a high pressure, so your population can recover a bit. But in any case, it's sustainable, but not in the long term. Okay, this is important for the yellow part, and the red part is not sustainable in the moment and either in the long term. So the ideal point for a management perspective, well, I'm going to show you here, is the center, because you're getting the most amount, which is the maximum sustainable yield, but your population remains stable. So these are we wanted to apply this theory of the stock assessment and the fisheries sciences to our scenarios in relationship with the trends that I showed you before. The idea was to see with these code plots what were the states of our stock in the different scenarios, comparing a population without variability and with variability, and comparing under different efforts of fishing. So what you can see here in the case of the individual, when there is individual variability, we saw that there were more biomass, there were higher catches, higher yield, and you can see it here that in the efforts 1, 2, and 3, that are the lower efforts, the population remains in the green zone. That is sustainable, and the population is even growing. It's allowed to grow. The higher efforts, of course, imply that it's easy to go to the overfish area. And in the case of when there was no variability, what we see is that generally in all the scenarios, the trend was to go to the overfish area. And we don't see a population that stays in the sustainable and growing or even in the maximum sustainable yield after the 100 years simulations. Each of these points is every 10 years where the population is, and it starts here in the black dots, and then it starts. And in the case of there was no variability, the dots tend to go to an overfish state under the biomass you need for the maximum sustainable yield. In the case of when we had individual variability, we had a maximum sustainable yield that was 11% higher, so we could get more fish, more catch, and the population would be stable. But it is also enforced by the fact that the population, so the biomass you need to keep that maximum sustainable yield, was 50% lower in the case of population when you have the individual variability. So you need less amount of fish to keep the same amount of catch, of yield. And well, this is related to the trends I was showing you. And you can see, for example, in the scenario 3 and 4, you have the higher catches, the higher yields, but you are fishing more. And the population is quite stable. So these two plots are meant to be observed together to find all the information. And later, what we observed was how the individual traits in the population were varying across our 100-year simulation. So we mentioned that we have a population. In this case, we are thinking the population with individual variability. And we start with a population that has a certain range of values for the chronotype, the home range, or the growth rate. So this would be, for example, we start in 10 years, when the simulation starts. And we can see that the trends over time is that this range of values for these traits tend to become narrower and to smaller values, to smaller. So this has to be, if you remember that the assumptions we made was that a higher home range, higher chronotype will imply higher vulnerability to fishing or to dying by natural rates, but especially to fishing mortality. And you can more clearly see this when you compare different. We were seeing the scenario 3, so the middle effort, let's say. If you compare with the scenario with the fishing effort is lower, you can observe this trend, but it's less intense. In the case of the highest scenario of effort, especially in the home range, you can appreciate that is quite strong, the decrease. So this brings an idea that was already described for natural wildlife, which is the timidity syndrome that is applied for populations that are under pressure, human pressure of hunting or fishing. There is a selection of the individuals, and the outcome is that the populations will become or will be formed by individuals that are more timid. So they have a smaller home range, they move less, they are less bold. This is something that has been observed in nature and described also for marine species and for the raor. And in the model, as we have parameterized, we can observe this process too, which is pretty interesting. And well, with this, we can get some conclusions that I have already mentioned through the results, which is that this is a first step, in implementing the individual variability in a population dynamic model for this species or for marine species in general. And we could see that incorporating the variability seems to make the population more resilient and allows to obtain a higher biomass in the overall population and also a higher yield, so you can fish more over time. That's important. And well, this individual variability also reduce the likelihood to reach the overfishing in the context of the same fishing efforts or the overexploitation with an scenario where there is no variability. You don't account for it. So this has implications in terms of fisheries management to create strategies that account for this individual variability at the moment of to obtain better long-term outcomes. And it is good for the fish stocks, but also for the fisheries and in the fishing, let's say. And well, the future steps that can follow and that are already taking place for the model specifically, it could be the parameterization. We've been working with a lot of parameters that come from natural processes like the mortality rates, the growth rate, etc. And we have parameterized based on actual and empiric data, but there is more. There is a lot a lot more to know in terms, for example, of growth mortality rates. And for this, and as part of the project MetaRaor, there is already being done a study of before and after impact analysis that can provide, for example, information of the natural mortality rates and fishing mortality rates in nature by comparing scenarios of marine protected areas where there is no fishing at all, so you would only have natural mortality, and areas where there is fishing, so you can have a period where you study the fish before the fishing starts and after the fishing starts. And with the acoustic telemetry methods, you can follow the fish that you have in these areas and see how much are dying and for which reason. And it brings you a lot of information that will be useful for the model to parameterize it in a more realistic way, or for this species specifically, for the raw. And then the same with, for example, in terms of establishing what is the relationship between the home range, the chronotype, and the growth rate to make this assumption that we applied in this model the more realistic or more database possible. You could compare, given that you have an individual and you know its home range and its chronotype, analyzing, for example, the otholites would give you also information on their growth rate, and you could see how these factors are related, and that would be also very important information to add to the model. And then all of this I have been explaining is a scenario where we have a population isolated, but in nature is of course not the situation that we find. So the next steps would also try to apply a metapopulation concept and perspective, with different populations connected somehow, and with this transference of genetics or any other thing. That would imply, that would be useful also to incorporate socioeconomic factors in relationship with the fisheries and the stock assessment. For example, with the figures of marine protected areas, that could be could be working as spillovers of genetic diversity that could sustain the diversity of the individual behaviors that we have seen that may be good for the population in terms of sustainability and resilience, and also for the stock assessment and for the fisheries activity in terms of obtaining more yields and having a population more diverse and less timid. So that would be it. I wanted to thank to Pep and all the team for this time with them. It's been super nice and I've learned a lot. And well, I hope that you enjoy. And if you have questions, now entertain.
So question.
Yeah, well, thank you. It was super nice. In your models, like when you have the blue curve and the red curve, temporal evolution, it seems that your initial conditions are different between the same
Yes.
pressure scenario. So you have like the blue line that was like the population variability, the red line the population without, but your initial density of the population is different. So is this for a reason?
Yes, it's because to start where we start the simulation, okay, we part from, in the case where we have no variability, for example, I simulated the population. In both cases, I simulated the population without fishing at all.
Okay.
So the initial population wouldn't be something that I basically created by myself, but I let it to simulate
So
under only with natural mortality for a certain period of time. And then I let the population grow like in the model context in a natural way without fishing, and then I introduce the fishing pressure.
This is not like a real initial condition, but there's like a previous simulation.
Yeah, the population is made up, let's say. So I create an initial population and I put it into the model, firstly, only with natural mortality, and then I include the fishing mortality. And the reason why the initial conditions are different is because in the case of the variability, the individual variability scenario, the population was growing a lot.
Okay.
So I stopped the simulation at some point because my computer was a bit hot. And then with the scenario of population without variability, I let it grow 100 years, and I started from that point adding the fishing pressure.
Because I mean, if you look at the last simulation, or the last 3 essentially, it's essentially the same curve, just shifted
Also, sorry,
in space.
also
In
you have to take into
density.
account, sorry, to interrupt, that it is not, we are starting in the year 20. I am showing from the year 20 because because of the scale of the y-axis, you lose a lot of detail if I show you from the beginning, which would be like here, you know? So the population will start very high, and then you apply fishing, and they decrease a lot in biomass. But if I would show from the year 0, the detail in the differences of these two trends would change a lot, because would be less appreciable. But you are having changes of the order in kilograms, which is a lot for our, which is small fish, where you have differences of, you see here, from 2,000 to 3,000 kilograms. So it is a huge difference, and the idea was to make it clear.
Yeah.
So that's why.
Not just because usually these models are density dependent, so if you start from a lower population and the dynamic, you don't expect that the population is half to have a temporal evolution which is at half. So not just you could use algorithmic scale.
Okay. If you see the trend of the population only in the natural conditions, only with the natural mortality process, let's say, without the fishing, it grows, and it is a logistic function. My problem was that with my computer, I couldn't get to the end of the function for the no variability for the scenario with variability, because it was taking hours.
Just that it's a bit confusing.
I stopped at some point to it's a computational limitation more than other thing.
And in between this scenario from E1 to E6, where are we now? Like, what is one of those?
We don't know where we. We don't know yet that information. It would be some of the information that would be good to incorporate to the model.
I put that this parameter depended on the fishing pressure, which I understand is something that.
It's column. Let's say this is E1, so this is F1, F2, F3.
So do you know which is the fishing pressure that right now is?
But right now in the natural, no, because that's very hard to know. That's why they are doing this before and after impact analysis. They are bringing this information, trying to get this information, or the abundance of the stock and where is it. In any case, I don't know it. Maybe there are maybe it is known, but I don't know it. Okay. Would say so. Can get.
I was wondering what would happen in the no individual variability scenario. You would use low home range and low as it seems that it's, I mean, the trend leads you to these values anyway. Would they perform better or worse than
Well, the thing is that that's the scenario you are getting. Like, that's the scenario that this timidity syndrome is leading you to, a scenario where there is no variability of the fish. And maybe in terms of the model, because we are giving to the fishing pressure a lot of protagonism, maybe it would perform better. But if you think of a natural environment, the trade-off of these behaviors to become less diverse is not desirable, because at the end will have implications for the survival and natural survival if you have individuals with less home range and less and the different chronotype, and you are reducing the variability. So in model terms, probably yes. If you start with a population with very low scores for these traits, as we have parameterized it, the outcome will be better. But it's not the question we try to answer, I would say.
Then what is the point of comparing non-individual variability with individual variability?
The point is to see if there are differences in one scenario or another, and to demonstrate or reinforce the idea that populations where you keep the diversity in their behavior will be more resilient from a ecological perspective, but also more productive for the, let's say, socioeconomic perspective. It's okay? Did I answer? Okay.
Yeah, actually, I had a similar point, and I actually have to say I agree with him in the sense that it seems to me that if you, it doesn't quite show what you were saying, meaning that if you choose better phenotype, even without variability, you can outperform your your thing, because in fact the model, if you keep it in the range where it doesn't collapse, it seems to me that it's going towards a selection of a specific trait, which is the best trait for the specific pressure that you're applying.
Well, but what's the better trait? Because
Well, it's
in ecological
selecting the better trait by the pressure that you apply onto the.
Yeah, but you don't aim to that. You don't want that specific traits are being selected for external factors like fishing, for example, or human pressure, you know?
Okay, well, this is I guess is a philosophical question, but so in fact the other question that
sorry, maybe I can add to that other thing that you can observe is when you get these less these populations with less diverse, and they are being selected towards more timid, for example, individuals with less horn brains. This also affects the yield, the catch, so the productivity you get from the fishing activity from a socio-economical perspective.
Right,
I don't know if that
connects with the other question that I wanted to ask, because it isn't 100% clear to me that this is the case, meaning the following. If I understood correctly, what you have is you have that in the spawning part, the parents transmit their phenotype, let's say, to the traits to the offspring, right?
Yeah.
So I was wondering whether you have tried to include, or maybe it was already there, but some sort of noise in this transition.
Okay.
So that you can go from complete non-inheritance, you can just, in a sense, with one parameter, you go from complete non-inheritance to exact inheritance.
Okay.
So some sort of noise in that. And then in order to see how, if you, for example, have variability, but no inheritance, that would show you whether the variability plays an effect by itself without having a selection during the simulation.
Yeah, that would be amazing to
But it's
be
just
able to implement it. But
have you ever tried?
at this level, I didn't get into that.
Okay.
It's just I just made a pool. This survived. So these individuals survive. They have this pool of traits. So they get to transmit it to in a random way. They get to transmit it to the recruits.
Obviously, I've done a bunch of simulations for each case, right? And then other.
Yeah, and in different scenarios. Yeah. Lot of simulation. Is it clear?
Yeah.
Okay.
Thank you for the presentation. Following a bit on Medea, what she asks, and if I understand well, from 0 to 10 years, you have your model, your simulation without the fishing pressure.
But I did a initial simulation from the population without variability of 100 years, and for the population with variability of more. But it's not the part that is left in here. It's previous to start the 100 years simulations to compare the scenarios,
Okay.
under different efforts of fishing.
Okay.
So it's not the 10 years that you're missing here, what I meant. If it's that
This simulation, what we see here, the biomass evolution, it comes from. Sorry, I didn't understand.
Yeah.
It comes from what simulation? So there's
you have an initial population, and I started to simulate their evolution, their dynamics, without fishing pressure for 100 years.
Okay.
Then I stop, and I start applying fishing efforts, and then that is when this starts.
So that's what we see after the
100
years.
But what I don't show you here is the first years of these 100-second simulations with the fishing for a practical
Because there's a big drop in.
Yeah, exactly.
biomass.
Yeah, I started in very high biomass and it drops and then steadies. But to find the detail, the interesting details that we want to see, I just put it from the
Okay.
year 20. Sorry if it is confusing. I didn't know how to. Well, I could have shown the plot with all the trend, but
Yeah, no, you're right. It makes sense. Okay, now I understand this. So all the consequences there are because you put fishing pressure on your populations, in your model. And so you affect all of these changes, trends and differences. It's because of the fishing pressure. What's your first 100 years, sorry, of initialization without fishing pressure?
Yeah.
Do you have convergence of your model or towards the state of a stationary state? Or a natural variability that is without drift, without trends, to be sure that what you see here, it's you don't have trends or drift from the first part of your initialization.
in the case of the natural, in the case of the pollution with variability, I didn't get to. I mean, I get to as more or less steady, but I couldn't affirm that it is the what do you mean, no? The steady or stable with a drift, but in a linear way. But because I couldn't computerly, my computer was freezing. No, freezing. No, the other thing was warming, and I was like, okay, let's start at some point and compare, and then at some point I can see. But yeah.
Okay.
So that's
a bit complicated. If you do have
that would be the ideal. I know.
or drift before this, it's also mixed with the filter pressure consequences. So you might have here both drift or trends from the natural variability, and then
actually
that is maybe numerical.
Yeah, you can see here because it keeps growing. Even you apply some fish, it keeps growing. Maybe because this first effort was very low effort. So this would be maybe a bit similar to the scenario where there was no fishing. And you can see that it keeps growing. But the moment you apply a bit higher fishing effort, which is very low because I didn't put the exact numbers to not confuse, but at the moment that you apply a bit of more effort, it doesn't grow that much. So I would like to think that it's okay.
So very good. Continue things question. But then if the first 100 over here are the same between the 6 cases, why are they different initial conditions? Like, shouldn't they start from the same place?
Yeah, but
because the population is the same between E1, E2, E3, and then you apply a different fishing pressure. So why there is an initial difference between the cases? If the conditions in the first part are the same, and it's a deterministic at the moment because you don't have noise.
Yeah, the idea was to not to give the model or the population some time to grow by itself. Let's say, because if I parameterize it from the beginning, everything, and I say I have 1,000 fish, and they are like this, and then I have another 1,000 fish, and they are all the same. I am the way I'm parameterizing. I can determine what is going to happen if I initially apply the fishing mortality. So the idea was to let the natural process go on for some time. So the population is, I know it's not.
Is this time different between E1 and E2? This initial time?
No, the initial population is the same in every case.
Well,
Because you're missing the 10 first years.
You're not seeing year 0 here.
You are not seeing the year 0.
It's the same simulation between.
Yes,
But it's a noisy simulation, but yeah.
There is okay.
Sorry, I was
Yes.
understanding the question.
Then I should have.
Okay, no.
No.
I mean, all those plots, they start from the same value.
They start all from the same. I can show you if you want. I promise you I have it.
So it doesn't matter what happened before the 100 years then, because they stand in the same value.
Yeah.
They stand both lines in the same value for all the simulation.
No, the colors.
No, for each color. So the red one, all the red ones.
The initial condition for each line is different.
The initial
Because
populations
the previous simulation
are
In the
different.
previous simulation, you have the natural evolution only considering the personality difference
Yeah.
between.
The initial populations are
But
different
if
because
you
the initial
want to
population.
check the impact of fishing, as she said, if you start with the same number of biomass, then you are sure that the differences between the two lines are between
Yeah.
the fishing and of the other.
For the record, the initial population with no variability was lower than without variability. So it's not that I started this, it's not that the one that I have more biomass is because I started from a population with more biomass. To avoid this, in case it was a skew or something, a bias, my initial population of the population with variability, the total biomass was lower than the initial population of the population without variability.
Is there any current capacity in this? Because I mean,
Yes, there is, but for the no variability scenario, I could get to it. For the no variability scenario, I didn't get to it.
But how? Because I guess that
But
how do you get to this current capacity? How is in the model this current capacity implemented? Because it means
well, the
mortality-
reproduction model, actually the formula was not complete, but I didn't want to. Well, I didn't have time to put it full. The formula of reproduction is the Beverton and Holt. Sorry.
Okay.
Beverton and Holt recruitment model. It has a carrying capacity concept,
Okay.
and this is like the basic historically used model. But I added to this actually a factor to add some variability, so it is not so much deterministic, and there is some random stochastic effect. So instead of a curve just like this, you have a curve more like this, but the function is quite similar. So that's what brings you. There is a carrying capacity in the recruitment that limits the.
But not in the
growth.
mortality, for example. I mean, because I guess that also when the population gets very big, maybe there are not enough resources for all the individuals, so their state is worse.
That is messy, but that is supposed to be included in the recruitment parts and the recruitment models, which are very complex, and generally in fisheries stock assessment is not something that no one considers that it's accurate currently, and it's, well, models that try to describe very complex scenarios. So there will be always a lot of things missing, no? But the idea is that it is supposed to be included only in this. I know it's.
Okay, so I think it's 4-1. So